Well, I started 9 years ago in Salamanca, Spain. In Salamanca, myeloma is probably one of the diseases in which we paid at the time more research efforts, mostly by heritage because of our boss there at the time, Professor San Miguel, which has a long-standing career in multiple myeloma. So this is how it started 9 years ago in Salamanca and more recently during the last 3 years in Pamplona. Within Spain, myeloma is one of the top hematological diseases being investigated. Then once you started, I think it's really easy to fall in love and become very passionate about the disease. It's a disease in which, for example, there is more than 50-year research about the cancer stem cell, the cell that originates myeloma, and for which there isn't still an answer. Nowadays we know that every myeloma patient is preceded by an MGUS and eventually a smoldering stage. MGUS patients overt to myeloma at 1% per year rate, smoldering myeloma 10% per year, but we still don't know exactly which are the mechanisms that dictate whether a patient will remain progression-free for 20 years or by contrast if the patient will progress next year, despite of also intensive research. Then lastly, fortunately the majority of patients respond to upfront treatment, but unfortunately after 5-6 years they just become refractory to the majority of the novel agents. So this is really the driving force for our continuous effort and research towards improving the management of the disease. Well, I think that there are two particular levels. One is risk of transformation, and the other is the development of sensitive techniques to assess or to measure the response to therapy. The identification of biomarkers that can predict with a great accuracy the risk of transformation, for example from smoldering to active myeloma, has actually led us at the beginning to the identification of a subset of patients with high-risk smoldering myeloma, for which the probability of overting to active disease was quite high, around 50% during the first 2 years, 75% at 3-4 years. And this has in fact led for a pilot study, the first clinical trial that asked the question of whether or not there was a clinical benefit for early treatment intervention in these high-risk smoldering patients. Fortunately, after 5 or 6 years since we started that clinical trial, in fact, the Spanish myeloma group was able to demonstrate that there is, I would say, a highly significant clinical benefit in both time to progression and overall survival for high-risk smoldering patients being treated with early chemotherapy, challenged with early treatment intervention. The second is obviously the fine-tuning of response assessment, particularly the development of technology that allows to measure minimal residual disease. And we have been showing throughout the last decade that the assessment of MRD, minimal residual disease, supersedes conventional response criteria, supersedes the concept of complete response in myeloma, and could hopefully in the near future, we are counting on that, though this still needs to be further demonstrated, could not only potentially improve the management of myeloma patients, hopefully tailoring their treatment, but also help pharma industry, the regulatory agencies, to approve drugs early on, before differences in survival that can only happen perhaps after 8 or 10 years of follow-up, so that the drugs can be sooner made available to the patients. Well, myeloma is not an exception as compared to other solid and blood tumors in which there is clonal evolution over time, or I would say even more specifically clonal selection induced by chemotherapy. I must say that in myeloma, this pattern of clonal evolution does not happen to all patients, but certainly 30% or more once the patient evolves from a newly diagnosed into a relapse stage. This is already well known. I think that despite that, we still need more studies to fully understand what is the impact of genomic abnormalities at the time of relapse. I'm not sure if we truly understand or if we can compare what is the impact in newly diagnosed patients as compared to the relapse refractory. Do the same high-risk cytogenetic abnormalities should be considered as high risk in relapse refractory? Are other abnormalities that at upfront level are considered as standard risk may in fact behave as high risk in relapse refractory myeloma? I think that these are important questions that still need to be addressed in the future. Fortunately, in 2016 we have very sensitive, well-validated and easy to standardized methodologies or techniques that would allow for a very accurate and a very sensitive MRD monitoring, hopefully to the vast majority of myeloma patients. So in fact, to have access to these methodologies or techniques is no longer a challenge. This should be more or less available to every myeloma patient. Then what to use or how to use that information, that remains a challenge. This is, I would say, easy to understand, and the reason is because before really using the information to make a decision, obviously related to treatment, you need to collect that information in a prospective manner, hopefully within the scope of clinical trials. So I think this is the stage we are at the moment, trying to implement MRD in the higher number of clinical trials as possible to understand its exact role to help patients in the routine clinical practice outside of the clinical trials in the next years. Well, we have learned a huge deal, I would say, but well, you know, I'm a huge fan of U2, and U2 have this tune that's called I Still Haven't Found What I'm Looking For. So there has been a lot of research, but unfortunately the research turned out to be, or the results were not what the different groups were looking for. So many different groups and research effort has compared the clonal cell at the MGUS, smoldering, active myeloma stage, either in different series of patients, even in longitudinal analysis, in other words, the same patient at MGUS stage, then when the patient evolved to active myeloma. This comparison started at a very low resolution level, for example phenotype, then they evolved into FISH cytogenetics, even copy number, more recently mutations through next generation sequencing. And what all the research efforts have showed us is that from a genetic point of view, the cell is quite similar from a benign into a malignant stage. In other words, all the cytogenetic abnormalities that can be found in a patient with newly diagnosed active myeloma can also be found at the benign MGUS stages. And this is why we believe that most likely in parallel or in addition to the genetic features of the tumor cell, the microenvironment plays an equal, if not even more important role. The interplay or the communication between the myeloma cell and the microenvironment, stromal cells for example, but also immune cell subsets. We believe that most likely the impairment of patients' immune system may actually be contributing to the continuous expansion of the myeloma clone, which ultimately will make the patient symptomatic and therefore candidate for treatment. During the last years, we have learned that in fact, smoldering myeloma is not really genetically different as compared to active myeloma. What we did learn after the seminal work of Professor Bob Kyle in the Mayo Clinic is that smoldering patients can be stratified into low, intermediate and high risk of transformation to active myeloma. What we have demonstrated in the more recent years is that in parallel with solid tumors, early treatment intervention of patients with high risk of developing active myeloma may be beneficial for the patients. And in fact, this has translated into a new diagnostic criteria. Since 2014, the International Myeloma Working Group has updated the diagnostic criteria, and now there is a subset of patients that may not show typical CRAB symptoms, but have myeloma defining events, namely 60% or more of plasma cells in the bone marrow, 2 or more focal lesions by MRI, or a free light chain ratio over 100. These patients should be offered anti-myeloma treatment. I would say that there isn't yet a significantly solid amount of data that can give you an honest answer to that, I would say, very important and challenging question. My personal perception of the field is that already at the smoldering stage there is a lot of clonal instability, most likely with more than one clone coexisting in the patient bone marrow, and upon transition from smoldering to active myeloma, you might have expansion of one of those clones. Though this may not be the case to all patients. In some patients you might have an expansion of more than one clone. I would say that in another fraction of patients you have an expansion of only one of the, usually, and this has already been demonstrated, 5 or 6 clones that coexist in the bone marrow of a myeloma patient. It's quite fascinating, and I would say that with the exception that both diseases have the plasma cell as the hallmark, as the tumor cell, I would say that everything else is rather different, and it's quite fascinating and should be a research, it should be a major research effort, because both diseases have the plasma cell as a common denominator. Nevertheless, for myeloma we know now a huge deal about the biology, pathogenesis of the disease, but that is clearly not the case for patients with AL amyloidosis. And I would really urge the scientific community to pay the same research effort in this disease. 2 reasons. One is because a subset of patients that might be diagnosed later on or not as early as they should be, and they have cardiac involvement, outcome for these patients is still very dismal. This is an unmet medical need in AL amyloidosis. The second is the potential likelihood of curing the disease. If we think that the tumor burden in AL amyloidosis is remarkably lower as compared to myeloma, if effective drug combinations are developed to this disease as they have been in myeloma, then we can foresee that after significant tumor depletion as we are seeing in myeloma, actually a fraction of patients with AL amyloidosis could be potentially cured. I think this is also something that should be further investigated in the future. it's a fascinating area. I think that we should all feel very, very grateful by living in this era in multiple myeloma. We have now very effective treatment strategies. We have now amazing technologies that allows us to understand better the biology of the disease, and we also now have very effective techniques that can allow us to measure the efficacy of treatment, of response to treatment. I think that all these combined should really allow us to move towards a much more personalized medicine in myeloma. So I think that that is one of the things I would be more excited towards the future. This applies, for example, to a better understanding of the cellular biology towards a tailored immunotherapy for myeloma, better understanding of cellular biology to discriminate between benign and malignant clones, and to understand how will this impact in the patient outcome, ultimately trying to prevent either under or over treatment in order to maximize life expectancy in myeloma.